Okay, greetings, CAMBAM, good afternoon. Thank you for coming to my talk. Hopefully it will be somewhat enlightening to you all, or tell you a little bit at least about what I've been working on for the past while, and hopefully it can be applicable to what you do and at least how you think about neurons and how neurons work. So it'll probably be a little bit different from the last few talks, which have just been mostly about statistics. I'm going to talk a little bit more about single neurons and sort of how they take an input and give an output. And I'm calling this all nonlinear systems identification, but just because being nonlinear is a little bit vague, you know, there's lots of ways you can be nonlinear. I'm going to actually spend a lot of the talk talking about linear systems analysis. And then we'll see kind of how that can fail and why you would need to go nonlinear or think about it in terms of being nonlinear, because it's not linear, which is kind of what that means, I guess. So we're just going to dive right into some linear systems analysis. And so first, imagine a neuron in your mind. It's going to get some kind of input, which we call the stimulus, and it's going to give some kind of output, which we call the response. Usually the response is a sequence of action potentials. You know, maybe you can actually measure the voltage, the membrane voltage, but just worry about the action potentials here. So the output is zeros and ones, the action potentials. And then you would, filter your action potentials to get some firing rate estimate, which is supposed to just tell you the number of spikes per second at every instant in time, right? And so, although neurons are inherently very nonlinear, that's what makes a neuron a neuron. We're going to imagine here a special case of a situation where this neuron, it's receiving zero stimulus, so the stimulus value is zero. It's not doing anything, but it's still firing action potentials. That's kind of important because we know neurons have this threshold where maybe they're not going to fire action potentials if you're below there. So let's just assume we're above the threshold. The neuron's just firing action potentials. Nothing's really happening, though. There's no stimulus. So the firing rate is essentially constant, but it's kind of noisy. Okay, and so then the first thing you do is you apply a stimulus. And to start things off simply, we're going to start with sinusoidal stimuli. So these are just sine waves. This one, let's pretend it's 2 hertz. And so the stimulus is clearly a sine wave. Now you can see the spike train. The spikes are a little closer together in some spots and a little further apart in other spots. And so when you filter that spike train and get a firing rate, clearly the firing rate is higher in some spots and lower in other spots. And because I've made this example very clear, it's pretty obvious that it's modulating like a sine wave with the same frequency as the input, perhaps a different amplitude and a phase shift. In this case, you can see that that's a sine wave, even though it's a little bit noisy. But really what you would have to do in many cases is you would have to try to fit a sine wave to the response. It might not be as visually obvious that, yes, that's a sinusoidal response to my sinusoidal input. So you actually have to do like a fit, maybe call it a linear regression. Maybe not. And so once you do, the red line there would then be the sinusoidal fit, which is the same frequency as the stimulus. And then you can kind of just line them up here, and what you really want to compare is the amplitude of the stimulus, peak-to-peak amplitude, and the amplitude of the response. And then perhaps, a shift in time between the peaks of the response, right? So the black is the stimulus, the red is the response. We've got their amplitudes there labeled, and the shift in time is kind of labeled there as well. Hopefully that's clear. And so then to describe this transformation, this linear transformation from the stimulus to the response, we call it sort of the gain and the phase. So we've changed the gain by making the amplitude bigger or smaller. In this case, it's bigger. The gain is defined as the ratio of the amplitude of the response divided by the amplitude of the stimulus. If they're the same amplitude, the gain is 1. And the phase shift is, you assume that the time from one peak to the next peak, capital T, the full period, is essentially 360 degrees, like in the circular statistics talk we just saw. And little delta t is just going to be the amount that you've shifted out of that full phase, right? So if you just say the percentage of the period, full period is 360 degrees, so this is going to give you your phase shift in degrees. Personally, for these intents and purposes, degrees I find somewhat more easy to use, but everybody knows that, pi by 2 is 90 degrees, or sometimes you get confused and think it's 45 or whatever. But don't worry about that here. So, okay, so we do this. We've done it for one stimulus, which is a sine wave at 2 hertz. But then, of course, you need to do this for sine waves at every possible frequency, or at least enough frequencies that then you can make a plot where you plot the gain value at each frequency and the phase value at each frequency. So maybe you're first going to start off with a few frequencies and you're going to plot them, and maybe you can fit a nice curve to it and say, okay, I pretty much understand what's going on in between those other frequencies, so I'm not going to worry about it, and that's going to be good enough. Or maybe you're like, we need to get more points at these high frequencies and find out what's really going on there. I don't believe it, you know? So you do as many frequencies as you can, and you plot the data. So now we have the gain as a function of frequency and the phase as a function of frequency. And hopefully, if you're somewhat familiar with the frequency domain and you've heard of a Fourier transform or maybe a Laplace transform, but if not, don't worry about it. You can just Google Fourier transform and come to me with your questions. Don't worry, I'll help you. But so what you want to do is you take the gain and you take the phase and you put them together into a complex function we'll call capital H, which is a function of frequency. And it's really like, imagine if you first have some complex number, you can take its magnitude and its angle, phase angle. That's what we have here. We measured the magnitude and the phase angle. That's the gain and the phase. And now we're putting them back together into a complex number. And so why would you want to do that? Well, this is the magic trick here with linear systems identification is this big capital H guy, H of f, is going to be essentially your neuron. And if you've done this at all the different frequencies, afterwards you can just take some arbitrary stimulus and you Fourier transform it. So you have capital S as the function of f. My laser pointer's not working. And so then you can just multiply S by H, and you get your output R. And you just had to multiply the input by the output, and you get the response. But it's in the Fourier domain, so then you have to just Fourier transform back. And really, this would be equivalent. Instead of having this complex gain and phase transfer function guy, sometimes you've seen some like wiggly little time domain filter. We're going to get to that later. This is essentially the same thing. If you Fourier transform your transfer function big H back into the time domain, you can do the same thing. You take it from the stimulus to the response, but then you have to do this convolution integral, which is a little slightly more complicated than multiplying maybe, but then you don't have to go to the frequency domain and back again. So it depends sort of on the situation you're in, but this is mostly just to give you an understanding of what linear systems analysis means. And now we're going to find perhaps an easier way to do this instead of using sine waves at all individual frequencies. Yep. Yeah, exactly. Yeah. Because it's not a straight line on the gain curve? Well, so actually, in this case, the fact that a system is linear doesn't mean that the gain or the phase curves happen to be straight lines. They can be as curvy as you want. The fact that really makes it linear is because this is just multiplied by h, and it's not like h squared or h cubed or anything like that. And so imagine this is like x, you know, so we're doing like it's kind of like a Taylor expansion where we're only keeping the x term and not going up to x squareds or higher things like that. So right now, linearity is not reflected in the straightness of lines. But in a couple slides, we'll see some lines where if they're not straight, that does mean that they're not linear. It's not really that the gain is linearly related to the frequency. It's that the response is linearly related to the stimulus. And the gain just tells you how that relationship works. Yeah, that's another test of linearity, is that the response has to have the same frequency content as the stimulus. If you put in a 2 hertz sine wave and you get like 4 hertz and 8 hertz and 16 hertz out, then something's nonlinear. And so we'll get to that. Yep. Yeah. It just tells. Right. And so it's just because if you have a linear transformation, you know, y equals ax plus b, the x is like your sine wave, but we're only just shifting it in time and scaling it. So there's only sort of, that's a linear transformation. Yeah? That's another nice property of linearity. Superposition, yeah, that just means, so if I have one stimulus, y1, and I get a response, or x1 gives a response y1, and if I have another stimulus x2 and I get a response y2, but then if I just combine those two stimuli into a new stimuli, its response would just be y1 plus y2 if the input was x1 plus x2. So that would mean the system's linear. And if that gets violated, it means it's nonlinear as well. So if the frequencies change, that means it's nonlinear. If the superposition fails, that means it's nonlinear. Eventually, we'll see an example where if a curve is not a straight line, then it means it's nonlinear as well, which is at least more intuitive. You think linearity means something is a line, but here they're all sine waves, which are not lines at all. But we'll get to something like that. So the main irritants with this method is that you have to apply stimuli at each frequency, which is time-consuming. So what if we just use, say, a white noise stimuli that's a Gaussianly distributed variable? We don't need to test it. Just assume that it's Gaussian. Generate it yourself. Make sure it's Gaussian. But let's say it's got frequency from 0 to 20 hertz. So now instead of doing all those individual sine waves, we just present the white noise. It has all frequencies in it, and we'll get a response, which will be a spike train. And of course, you can filter it again to get a firing rate response. But now it's really tough to just look at it and say, oh yeah, that's clearly a linear transformation of the stimulus. Can't you see it like wiggles in exactly the same way? No, you can't tell. You have to do some analysis to check, right? But what I want to show here is that you can use this type of data to create the transfer function just like before, this h guy. And so if we assume this kind of little relationship between h and s and r, these capital S's and R's mean we're in the frequency domain instead of the time domain. And then I got this extra little trick here where I put a star by some of the capital guys, which technically means complex conjugate. If you know what that means, great. If you don't worry about it too much. But what we get here is this relationship where the transfer function h of f is equal to s star r over s star s, which now I'm going to tell you what those things are. These are actually, s star s is actually the autocorrelation function, so S correlated with S, but we've got this extra little lag tau in there. But then we Fourier transformed the autocorrelation function. And so what that actually is the power spectrum of the stimulus. And I'm going to call that PSS, power spectrum of the stimulus. You could also have power spectrum of the response, which I would call PRR. And then we've got this extra nice little object here called PSR, which is the cross spectrum, or if you want, you could call it a cross-spectral density, but I don't really mind if you leave out the density word. Depends who you talk to, though. Some people get really upset. So just in case. And so let's say we actually do that. So we calculate these cross-spectrums, and I mean, we can have a whole class on spectral density estimation techniques. But let's say you can do it. You've got some lab code, so you calculate your transfer function h by just that simple method. And then if you took h and say, well, what's the gain? You take the magnitude. And what's the phase? You take the angle. You can plot those, and you're going to get back the same thing that you got before using the sine waves. But now you just use a big chunk of white noise that already has all the frequency components in there. You don't have to do each one individually. But of course, if you don't even know if you have a neuron isolated, and you apply sine wave noise, and you get some noise response, you can't really tell, like, oh, yeah, I've got a clean neuron there. So that's one of the advantages of using the sine waves to do it. So they're kind of similar techniques, complementary techniques. Let's go with that, complementary. So here, now just to sort of round out the linear component of the talk, we're going to talk about linear stimulus reconstruction. So there's another situation where you give the stimulus, you get the response, but as was pointed out before, really what the brain wants to do is it's going to get spikes into the brain, and it has to figure out what was the stimulus that caused those spikes, right? And for here, we're not going to try to distinguish a spider and a tower, but we actually want to reconstruct the entire time course of this whole wiggly stimulus as accurately as possible, right? And so now we're going to look a little bit more in the time domain instead of the frequency domain. And we've got the response R of t instead of big R of f, and the stimulus. Now this little s of t has a little hat, which means it's the stimulus estimate. And instead of having our filter called h, now we just called it k. I probably could blame whatever reference I copied this out of, spikes. But so now what you want to do is you want to say, well, I want my stimulus estimate to be the best possible. So I want the error between the stimulus and the stimulus estimate to be really small. So we're going to say, let's just set the derivative of the error equal to 0. Unfortunately, it's a functional derivative, so it's a little complicated. And you need to do a lot of mathematical trickery, which I'm going to leave as an exercise for the reader. If you want to try and get stuck, we could talk about it. If not, just look at this useful result. But so in many simplifying cases, if your stimulus is, white noise and you're very linear and everything, this optimal filter in the frequency domain happens to be PSR of negative f divided by PRR. And so you think, wait a second, that looks a little bit like the transfer function, only it's got PRR instead of PSS, and this weird negative sign there, which is true, and that's how it is. And so just to kind of complete the story, because everybody's probably heard of this special thing called the coherence, which kind of measures how well the system, the response, or the stimulus and the response are related linearly. If they're perfectly linearly related, you can go from the stimulus to the spike train. There's no noise added to your system. It's totally linear. You go back to the stimulus, and it should match perfectly. Then you would get a coherence of 1. And if you can't reproduce it at all, then you would get a coherence of 0. But one thing you can just notice, the coherence is actually PSR squared over PSS and PRR, which is essentially like multiplying your transfer function and your reconstruction filter together. It's got both of them in there, right? So the transfer function is like going from the stimulus to the response. The reconstruction filter is going from the response back to the stimulus. But the coherence, it doesn't really tell you about which direction. It just tells you that they're correlated, but it doesn't tell you that the stimulus actually caused the response or that the response caused the stimulus. But, with neurons, generally the stimulus causes the response. So it's not too much of a problem there, but at least, and then, you use these techniques, you can get the coherence, but the coherence is also related to mutual information. So that's another reason why it pops up a lot. And we're not going to talk about it anymore from here on out. I just wanted to kind of show you that nice little connection of these concepts. Okay, so now what are we going to do? We're going to look at an example near and dear to my heart from the vestibular system. So let's imagine you have everybody vaguely knows what the vestibular system is. There's these neurons in your brain which are sensitive to head movement, angular rotations, linear movements. So let's say we have a neuron that responds to horizontal yaw rotations, and we've got that neuron. Somebody's recorded it painstakingly, perhaps in the audience right now, and I'll just get the data and analyze it. And so let's say we've also already done that thing where we applied all the sine waves and we got the transfer function, so we pretty much know how the neuron works when it's linear. But so now we're going to give this fancy stimulus, which it has multiple frequency content, or multiple frequencies in there. It looks somewhat sinusoidal, but then the amplitude changes. So the amplitude is going to get so big that it's going to make this thing go nonlinear instead of just being linear, which we'll see. But so the black there is the stimulus, and the red is the firing rate response. I've already filtered the spike train to get a firing rate estimate. And you can see that they do look pretty similar. But, maybe not perfectly linear. Maybe it's slightly nonlinear. So we're going to test it out. And so if we think that it's linear, we already have the transfer function that we made using the white noise before. And so now we can just put the stimulus through the transfer function and get a predicted response. I can call this response the linear prediction because we just used the linear transfer function to predict it. And then I've plotted it in green here. And so you can see that the linear prediction, it does match the actual response reasonably well, especially in certain regions. You can tell which spots it matches well and which spots it doesn't, right? You can see that it's really going too low and a little too high with the linear prediction. And actually, these dashed lines here indicate 0, where you know that a neuron cannot have a firing rate less than 0. It just doesn't even make sense. So if you get that in your data, you know something's wrong. So that's a very good clue that the system is somehow going nonlinear. But because neurons often have this absolute refractory period of firing spikes, they can have a maximum firing rate as well as a minimum firing rate, which you can't go past, which is what the linear prediction here looks like it's doing. And so we'll say perhaps we need a nonlinear function as well to make the prediction more accurate. And so now my little top panel there is supposed to be a schematic where it shows first you put the stimulus through the transfer function to make a linear prediction. That's the linear component. And then we're going to put it through a nonlinear function to get a better prediction, which we'll call the nonlinear prediction, for lack of a better name. And so now we want to say, well, what nonlinear function will make this work so that we can accurately predict the response? And so we're going to try to estimate the nonlinear function from the data. And the way that we're going to do that is you take the linear prediction and the actual firing rate, the green and the red guys, and we're going to go through every point in time. And for each point in time, we get two numbers. One's the linear prediction, and one's the actual firing rate. And we're going to go down here, and we're going to plot them. And so you can see that even when the linear prediction goes very low, like minus 200 or something, the actual firing rate never went that low, and it never went below 0, actually. And so every point in time is going to contribute to a point on this line, and then we just sort of average all those points in bins here, and we get sort of the red points there are the mean with standard deviation or error bars. And so you can kind of see just from the red that it follows the sort of sigmoidal shape that we would have thought, because you can't have a negative firing rate, and there should be a maximum firing rate. And so you can play around perhaps with exactly which kind of function you fit to your nonlinearity. Maybe its shape is slightly different from this sigmoidal function that I've chosen here, but it's not going to for the purposes of illustration, that doesn't really matter. And sigmoids are fairly common, especially in engineering, and they're simple functions that capture the main important properties. So we fit the red data there to the sigmoidal line. And then what we can do is we take the linear prediction and we pass it through this nonlinear function. So then every time the linear prediction has a certain value, you just go on to the nonlinear function and you say, oh, I have a value of 300. Okay, so that's what the actual firing rate value should be. And so once you've done that, then you can get your nonlinear prediction, which I then plotted there in blue, and you can see it at least matches much better with the actual firing rate. And then if you want to check, is this really good or not, you can do the same thing where now you plot the actual firing rate versus the nonlinear prediction instead of versus the linear prediction. And we put that in this little inset up here, and you can see that it fits on a straight line. And that straight line means the system's linear. The fact that this is not a straight line means the system is nonlinear. So in this case, the linearity of the line corresponds to the linearity of the system. And one thing that's kind of important here is if the transfer function really accurately did predict your gain properly, then the nonlinearity should have a slope of 1 in the middle here because it doesn't change the gain, right? And the neuron fires spontaneously, so if there's no stimulus, it's firing sitting right here. And if you give a really small stimulus, maybe it just wiggles back and forth here, and so it stays nice and linear. But if the stimulus gets too big, it pushes it into the nonlinear regions, and you start to get some like clipping or cutoff and saturation. And so that is, and the R-squared values here just tell you that, look, the straight line fits the nonlinear prediction plot much better than the linear prediction plot. So we've seen a significant improvement in the model. And so that's great. But now probably the question or the concern that you all have is like, what the hell are you talking about? Our neurons don't fire spontaneously and just modulate nice and linearly like that. like, what, you know, usually your neurons don't really fire very much, and then if you find a stimulus that they like, they're like, whoop, and they start firing. So how is this model, is this approach going to be applicable for those type of neurons? So this is another technique that you've probably all, you know, read about in Diane and Abbott or one of those neuroscience textbooks. We're going to do the same thing. We apply the white noise stimuli, and we get the spike train, but instead of just doing the cross correlation or the cross spectrum and getting the transfer function, we're going to do a spike-triggered average, which essentially amounts to you have your stimulus and you have your spike train. You've aligned them so that, they match in time. And what you can't see here is that there's a little pink box that starts at this spike and goes back in time a little bit here, highlighting a snippet of that stimulus that just precedes the spike. Actually, I'll take a little bit that comes after the spike too, just for fun, but they're not showing up here, unfortunately. But does everybody get what I'm getting at with the spike-triggered average? Because my next slide here now, there's windows around every one of those spikes, and so we've highlighted a bunch of stimulus segments, S1, 2, 3, 4, 5, up to 9 here, only 9 spikes of this neuron. Hopefully you get more like 10,000 spikes when you want to do this, because 9 is not going to be enough to get a good average. You're below the threshold for good statistics with only 9 samples, seriously. You want more than 30, I would say. But so then, so these equations aren't very clear either. So basically what you want to do to make a spike-triggered average is you take every one of those little stimulus snippets, S1 up to 9, and you just average them. And that gives you, we'll call it like a filter H, but it's a spike-triggered average filter, okay? And it's going to look somewhat like the segments, but the average of them, there's going to be a lot of variability in there from noise. But what makes this a reverse correlation is if you think of the response as a sequence of delta functions, so it's just zeros and ones. If you actually took a correlation function, a cross-correlation, stimulus and response, that's the same thing because the response is zeros and ones, so it's only pulling out stimulus segments that are there. And so this is actually still a cross-correlation function. And so really, if you Fourier transformed that, it pretty much is your PS of R. I should have had an inverse Fourier transform sign there. Apologize. So it's really, you're still your cross spectrum, but it doesn't have that stimulus power spectrum in the denominator, so it's not technically a transfer function. So sometimes people will just add it. They'll be like, I'm going to normalize my STA by the power spectrum of the stimulus to make it better. Really, it's just making it, it's a transfer function, but now your system is nonlinear. So why can you make a transfer function if it's nonlinear? Well, you can't really, but you can kind of, and we'll see why. So basically now, instead of just putting it through the transfer function, you put your stimulus through your spike-triggered average filter, and you get your linear prediction. Some people call it a projection, you know. If you get that, don't worry about it. It's pretty much the same thing, it turns out. But this time, you really need this nonlinearity. It's not like the stimulus, the linear prediction is accurate, except maybe it's wrong in a couple of small regions in the extremes. This might not be right at all. And so now you need to go back to your firing rate estimate that you got at the top, or you could do it with the spikes alone, but that's a little more complicated. And so now we just do the same thing to plot the nonlinearity, where we say at every point in time, what's the stimulus projection value, and then what's the firing rate that corresponds to. But so now, when there's no stimulus, perhaps the stimulus projection value is 0, the firing rate is 0. The neuron's not firing at all. And so you actually need to get, you know, the stimulus projection value needs to get up higher to get some firing, right? And then you know, the firing rate may go up, but it should eventually saturate because they can't fire infinitely fast. Anyway, but the other main thing that separates this from the transfer function is that the transfer function, the gain of the neuron was contained in the transfer function, and it would be the amplitude of this little filter guy. And then the nonlinearity had a slope of 1 because the gain was accurate. But here, this is not the case because it's not in a linear response regime here, right? So here, the slope of this thing will actually tell you the gain of the neuron with respect to that stimulus feature or projection or filter value. Does anyone have any questions? Reasonably clear? Following me somewhat? So now you need the nonlinearity, but here these nonlinearities are still kind of boring, right? They're just mostly...
Just to clarify, so the spike trigger average,
Yeah.
this is the filter that you're extracting from your spike trigger average. This is a linear filter. Is that right? Or is it a non-linear filter?
I would call it a linear filter, but I'm not...
So you don't have the non-linearity in it? So
Yeah.
you have the stimulus, projection, and then you simply add the non-linearity.
Exactly. That's right. Right. So we kind of call these like linear nonlinear cascade models because you first go through a linear box, then you go through a nonlinear box. And so you can kind of decompose it into linear nonlinear components. Maybe if you get to very highly nonlinear systems, you can't even do this kind of thing. Maybe this is only applicable in special cases, but we'll at least see one more kind of nonlinearity or a way to make it slightly more complicated. If I still have time, hopefully. Okay, so now we're going to switch systems. This is now no longer from my own experience as much as just from a paper that I read that seemed cool. So rat whisker system, the whiskers move, and then the neurons in the cortex, the firing rate encodes the whisker position or velocity or acceleration or something. And so what I've shown you before, with the spike-triggered average, we extracted one filter, which was your spike-triggered average filter. You can use a slightly more complicated technique where you take all those spike-triggered segments, put them into a covariance matrix, diagonalize it. It's called spike-triggered covariance. And you can get multiple filters out. And if you are good with linear algebra, you look at the eigenvalues and see if the eigenvalues are good. And you get two filters this time, or eigenvectors, if you want to think of it as a projection in subspaces. But if not, just imagine we have two filters here. One of them is stimulus position, and maybe one of them is stimulus velocity, so like the derivative of the stimulus. And now, every time we have a stimulus segment, we can see, okay, so it lies somewhat in the position direction and somewhat in the velocity direction, or maybe in the negative directions. So every stimulus segment looks sort of like a combination of these two filters. Okay? If that doesn't make any sense, don't really worry about it too much. But so if we go back to our imaginary example where you give your neuron a sine wave, here we're looking at repeated trials, right? And so you can see that it's firing more at the down phase of the sine wave. And so now let's imagine what happens when we do this spike trigger analysis on the neuron. So we've got, basically what's going to happen is we've got two filter shapes now that the stimulus is going to be filtered through both of the filters separately. And then they're going to go through a nonlinearity. And in this top little panel, we just show one nonlinearity. But because there's two filters, we should really have two nonlinearities. And instead of having two separate nonlinearities, we make a sort of two-dimensional nonlinearity. So if there's no stimulus, projection value on filter 1 would be 0, projection value on filter 2 would be 0, and perhaps the firing rate will be 0. And then if, you have some kind of stimulus, you filter it through one, and you filter it through the other. And so you take both of those numbers to go into your two-dimensional space, and then you look at the actual firing rate response and say, what's the firing rate there? And that defines the color here. So this is like a two-dimensional nonlinearity. And then these guys on the side are just saying, well, if we project the nonlinearity into just one of the dimensions for each of the individual filter components, this is what they look like. And so obviously, the gain or the strength of the response for this dimension or this filter is much stronger, and then the component for this filter is very weak because the slope here is not as high and strong, right? But these nonlinearities have the same basic shape or structure as the previous ones we've seen, where in one dimension there's no firing and it's flat, and in the other direction it starts to fire and you get a slope going up, right? So it's pretty much the same as what we already saw for the previous one. But then what if you encountered a mysterious neuron that responded like this to a sine wave, where here the mean firing rate never really seems to go to 0, so you might say, oh, geez, this neuron's pretty linear. But it's not actually. And one of the clues, if you like the frequency content of your responses and such, the stimulus has one frequency component. I don't even need to tell you what it is, but you can see that the response on the right-hand side has double that frequency in the response, right? Because it's responding twice as often. It's responding when the stimulus goes up and when the stimulus goes down. That's weird. Why would it do that? So let's do this analysis and see what we can find out from that. And so here we basically use the same filters, so we're not changing the filters. But now when you make the two-dimensional nonlinearity and look at the projections in the same way, you can see you get this crazy U-shaped nonlinearity, which we could call a squaring nonlinearity perhaps. But if you think of, again, like your Taylor expansions, you got like 1 plus x squared. So these simpler nonlinearities on the left are basically just like a line, which is like an x term with a slope that says the gain. Then these U nonlinearities, it's really like your x squared term in your Taylor expansion, right? Because it goes up on both sides. And so I would argue perhaps that this is even more nonlinear because it requires a higher order term in your Volterra expansion, perhaps, which hopefully Theo will explain to me later. So this is another type of nonlinearity that's sort of fundamentally different from the first type. And then, of course, there's other crazy things you could do. This is just to give you some more examples of the vast nonlinearity zoo out there of creatures that you might encounter. So another thing that they did with this rat whiskers is they took all the whiskers and they put a white noise on each one. But they still only recorded the neuron connected to one, right? And then when they calculated the nonlinearity, they set whether the noise stimuli, whether they were correlated, so they were all doing independent noise, or whether they were all doing the same thing. And then they actually found that the structure of the nonlinearity changed as you changed the correlation. So that makes it even more nonlinear. If the nonlinearity itself is changing with the stimulus, you probably need even more higher order terms than that to explain why it's changing. Right, and so that's just another possible thing you can do with this is the main idea with the linear nonlinear model that when you use the white noise to make your nonlinearity and your linear function is that you want that to be general, so then you can use any stimulus and you can still predict the response well, which that would mean that the nonlinearity doesn't change when you change stimuli. But in this case, it does change, right? So if you measure the nonlinearity using uncorrelated noise, and then you want to predict the response when there is correlated noise, it's not going to work. And so it's actually more complicated than that, right? And so then you need to map it out, the nonlinearity, with the different stimuli. And now just because I've given all these examples using sort of nice one-dimensional stimuli from the vestibular system and the rat whisker system, but perhaps you all work in the auditory or visual systems, which are so much more popular. And you're like, wait a second, my stimuli are two-dimensional. This doesn't make any sense. But you can still do it. And so in this case, this is a two-dimensional, so you have an auditory stimuli, you take a spectrogram of that, so you've kind of got a two-dimensional stimulus, which is a spectrogram. But you can still just do the spike-triggered average where you take little pictures in 2D, and you average them all, and you can get a spike-triggered average, or a STRF if you're in the auditory field. And you basically do the same thing with it. And you can get the same types of thing. You can do one-dimensional nonlinearities that just are flat and then go up. Or you can do two-dimensional nonlinearities, and you can get this other U-type shaped nonlinearity. And in this case, in their two-dimensional guys, on one axis it's the one type, and then on the other axis it's the U type. So you can even have the combination of both. It doesn't have to just be as simple as the examples that I chose. And actually, this is the last thing that I have here, but now you just need to step into your imaginatarium and imagine your neuron is only responding to towers and not to spiders, or is only responding to Robert Downey Jr. and not another Marvel superhero, you know. And so that would be a highly nonlinear neuron that is only responding to one or the other. And so perhaps a simple linear nonlinear cascade is not going to be enough to fully capture such a highly nonlinear system. But one thing that you can do is, because these things are so modular, you can make it like linear, nonlinear, linear, nonlinear, and have a little chained cascade. Or you can put them into parallel and have like many linear filters that all converge down into a nonlinearity. And you can build that up, you know, adding layers, and because it's all kind of modular. And hopefully, eventually, you could reproduce these highly nonlinear cortical neurons. But in those cases, perhaps this technique isn't always as useful. But if your system is kind of mostly linear and a little bit nonlinear, then this can be much more useful. And of course, none of this analysis is nearly as simple as I've tried to make it look. There's all sorts of little caveats and pitfalls and difficulties. And if you want to talk about those things, we can. But I tried to just sweep it under the rug to make this at least seem reasonable and coherent in this amount of time. So, did we make it? Made it in time? All right, thank you. All right, I'll work on my conclusion slide for next time. If anybody has any questions, I guess we have a couple minutes to handle questions. If not, yeah, Leonard?
So if I get out the neuron and nature neuroscience papers that people have to destroy all the method.
Right.
So the idea is really that you want to go through the process of putting white noise as an input to your system.
Yeah.
And it's a very effective method to arrive at something like a lookup table, how you go from the initial linear prediction to what the actual response is.
Right.
So I can think of this as like the construction of the lookup table.
I guess so, yeah. I mean, the nonlinear function is kind of, yeah, you just look up what the linear prediction is, and then it tells you what the nonlinear prediction is. Right, yeah.
When you present the nonlinear function like this sigmoid,
Yeah.
an assumption that
Right.
conversion was a sigmoid. But if you do like simply lookup, then it's
Yep.
not parametric, so you know it's nonlinear, but you don't make any assumption about the underlying function.
Right, yeah. So I averaged it, and then I fit it to a sigmoid, which is a simple, you know, parametric representation. But what you could do is just take all your data and completely make a map called, you know, the conditional probability of P of R given S, and just use the data and say, well, I don't know what function that fits. I'll just use the data. But, you know, if you can get a function, then it makes it even cleaner, maybe. Yep. Yep, you can do that. Okay. Yeah. Say again? Yeah. So what are you, an engineer or something? Yeah, because like, I know, like, technically there are some caveats there. Like, how do you, if you made the gain bigger, then the slope of your nonlinearity could just be smaller, and you'll get the same result. And if you made the gain of your transfer function smaller, then you just make the slope of the nonlinearity bigger, and you still get the same result. And so it's kind of like there's no unique representation there. And you could tell your computer, say, I want you to find the representation that the nonlinearity has a slope of 1, and then just do it iteratively to figure it out. But the other way around that is if you use a very low amplitude noise and you know it can respond linearly, you get that accurate linear part, then you use the bigger noise that goes nonlinear. But if you just started with noise that was already nonlinear, then it's really hard to get the linearity alone, right? So I think that's what you're kind of referring to. But you definitely can have other systems where you put a nonlinearity, then the linear part, and then another nonlinearity.
Implicitly, if you start with a nonlinear,
Yeah.
you implicitly have a linear part which is equal to 1. Am I right? If your first block is nonlinear, your first block can be linear and nonlinear as well.
Mm.
Like, it doesn't have, your first block does not
Right.
can happen. The first moment doesn't have to be linear and followed by non-linear. Non-linear can be inside your first block.
So is it, I've got a question for you then. Is there some parts where you can't separate the linear and nonlinear part into two blocks? You can, but you can just leave them combined in one. Okay.
It's not that they act on the input at the same time.
Right.
Their outputs are combined.
Okay.
And then you can pass it through like a non-linear to
Right. Okay.
but the filters, the linear and non-linear filters are applied at the same time.
Okay. Okay. Excellent. One more question. Yeah. What's the Hammerstein? Then nonlinear? Oh. Okay. You just force it to be one. Okay. And so it's a Wiener system if it goes linear, nonlinear? Yes. All right. An engineer told me that once. He was really excited. He's like, wow, you have a Wiener system. I was like, oh. Thanks. Thank you.
So if you go back to, I was part which you presented PCA,
Oh, yeah. I really glossed over that pretty heavily, eh? But I totally skipped over the details there.
Yeah, I was just curious. I mean, does your filter? I mean, there's a notion of orthogonality here. So your filters needs to be your stimulus needs to be orthogonal. I mean, you need to.
Your filters. Right. Yeah. Yeah.
Stimulus is only white noise.
Yep.
And filters ends up being. Oh, from the PCA. That's what we
Yeah. The eigenvectors kind of. Yeah, that's exactly it. The stimulus has to be Gaussian, and so if you try to use, like, you can do this using sinusoidal stimuli or other stimuli that are non-Gaussian, but then it's a little, just interpreting everything can be a little trickier, and there's more issues, like all those little caveats start popping up. So if everything's Gaussian and linear, everything's easy, but.
It just, do you start, the way to do that, so you have your input supply, and do you like generate the population of filters that you pick?
Nope.
Then you do the PCA population data.
Nope, you do your spike triggering, right? So every spike gives you a little stimulus snippet. That's your population, and you do PCA on that. So you put those into a covariance matrix, diagonalize, and take out the top guys. There's other tricks you can do, such as, take the first 5 PCA, project everything onto there, and then do it again. So it's like you're filtering out some noise, perhaps, if you do that.
Your vector that you feed is
The spike-triggered segments, yes. So, yeah, exactly. And so you could call it, you could call it the population of spike-triggered segments, like the spike-triggered ensemble. And so then you can do a covariance analysis on that, or you can just average it. Yeah. But then, finding, how many filters are good and significant is you look at the eigenvalues, and hopefully, like, 2 of them pop up above all the rest. But usually you just get some, like, continuous, like, blur of crap. Like, it doesn't always work, obviously, right? And so when it does work, it's really nice, but when it doesn't work, you're sitting there and you're like, am I doing something wrong, or is this just not going to work at all, or should I keep trying or give up? I don't even know. So it doesn't always just spill out quite as cleanly as these toy examples did, but.
And when, if you go just back to the figure 2, so
Right here, yeah.
this is the angle, this is y.
So technically the stimuli is the same white noise, but now every time I can take a segment of the stimuli, and then imagine this is a vector. I just take a dot product between the stimulus and one filter, and I say, well, how similar are they? And I take a dot product between the stimulus and the second filter, and I know that this, the square of the 2 should add up to 1, so it should lie on the unit circle. The reason it doesn't is because some of it lies in the direction of all these other filters that we got rid of, so of course it's a little less than 1. but it's close. I mean, I guess these guys are problematic, but so that's why they call it like a projection. Each filter is like a dimension, and this is saying for every stimulus segment, which direction does it lie in this space? Is that somewhat clear? But you can also, you don't have to think of these as vectors, you can just think of them as filters, but it's essentially doing the same thing. If you put a stimulus and you filter it through a filter, you're essentially taking every segment, projecting it onto the vector at every step in time. And so you can kind of think of them as the same thing. But yeah, this technique is a little bit tricky and could have made a whole talk just about that, but good? All right.